Good morning everyone, myself Shlok Keshwani, and today I am here with the part 4 of chapter cells, the building block of life. In this we will be covering our leftover topics with some practice questions and handwritten notes. So our this topic was left. So here different kind of solutions are given. First one is isotonic, hypotonic and hypertonic. In isotonic, the solution concentration means the solute concentration outside the cell is equal to a solute concentration inside the cell. Isotonic solution. Hypotonic solution, if we talk about, then here the concentration of solute है ना आउटसाइड का सोल्यूशन इज लेस देन द इनसाइड का सॉल्ट का कंसनट्रेशन एंड इन हाइपरटोनिक द कंसनट्रेशन outside the cell is more than inside the cell. Okay. Then this exercise we discussed, and yes, this threads of curiosity was left. So in 2010, scientist named J. Kerrick Venter and his team made an important discovery in the field of synthetic biology. Fine. They first studied the complete DNA sequence of a bacterium called Mycoplasma. Mycoplasma is a kind of bacteria, prokaryotic cell. Prokaryotic cell. Fine. See, in this chapter, many a times you would have heard the term of this prokaryotic, eukaryotic. So let's now talk about the difference between. Okay, so if ever you are hearing this term eukaryotic, देखो first you need to understand that eukaryotic, the meaning of these words you will get to know when you will break these terms. Fine. So here how will we break? You wait sorry. U and karyotic, they will be broken. Here U means well defined and karyotic or karyon means nucleus. Actually to EU means true, but for your understanding I am telling you well defined. So what is the meaning of eukaryotic? A cell, a kind of cell which contains a well defined nucleus. But instead, if you're talking about prokaryotic cells, again you have to break the term pro and karyotic. Here again karyotic will mean what? Karyotic का तो meaning will not change, it will remain nucleus only. Nucleus. And pro means primitive. Primitive means not well defined. Understood? Means in eukaryotic a well defined nucleus is there, and in prokaryotic a well defined nucleus is not there. That's why all the bacterial cells, जितनी भी आपको bacterial cells you will find, they come under this kingdom, and this kingdom of bacteria is known as kingdom Monera. About kingdom Monera, Protista, Fungi, Animalia, Plantae you will read further. Classification of living world, and you will also read out Archaebacteria, Eubacteria, that is another kingdom classification. So is this clear now? Okay, then let's come back to our topic. Here bacteria means mycoplasma. I told you that all the bacteria come under which kingdom? Monera. Yes, Monera. So prokaryotic means Monera, and all the bacteria are prokaryotic, means they do not have a well defined nucleus, है ना? तो bacterium is singular, bacteria is plural, are plural. Bacterium called mycoplasma, myocytes, using a computer programming. Then they chemically synthesized an exact copy of its DNA. मतलब इसका मतलब ये हुआ in 2010 J. Kerrick Venter along with his team they made an important discovery in the field of synthetic biology. Which biology? Synthetic biology. They first studied the complete structure, DNA sequence basically, of a simple bacterium known as Mycoplasma myocytes. Using a computer programming, with the help of computer programming nowadays you can even store DNA, which is biotechnology. Then they chemically synthesized the exact same DNA, so they did not, but the copy. Next, they took another closely related bacterium and removed its DNA, but kept rest of the cells. देखो. And next day, they did not take this exact mycoplasma myocyte, sorry. They took any other bacterium which was related to mycoplasma myocytes, and they removed its DNA but kept rest of the cell organelles such as cytoplasm, cell membranes, etc. Fine. Now they inserted the DNA of mycoplasma myocytes in a closed related bacteria. Fine. They inserted the synthesized DNA into the cell. After this, the cells started to grow and divide. When they inserted the synthesized DNA of mycoplasma myocyte in a closely related bacterium, the cells started to divide. This experiment showed that DNA controls the structure and activities of the cell. DNA का ऐसे यही function नहीं है to give out genetic information. Its function is to control the structure and activities of the cell. However, scientists did not create a completely new cell from scratch. Only DNA was synthetic. The other parts were same. है ना? So this was all about threads of curiosity. Let's read about how normal cells grow and divide. So this activity is that about what you have to do. You have to take a jar, fill it with plain water, like shown in this figure. Then after, you need to take an onion bulb scientific way में a bit बोला गया है, not proper. So, onion you have to take, and you have to arrange it in such a way that this base touches the water. Fine. In such a way that this base or those barely roots are immersed in the water. Fine. You have to leave this setup for how many days? For 5 to 6 days. Fine. After that, what will you observe? You will observe that these roots will start growing. These roots which you can see here, they will start growing. This will be your observation. Then after these freshly grown roots, you have to cut 2 to 3 centimeter. Here though I cannot measure digitally, but according to your assumption, you have to cut. And those 2 to 3 10 centimeter of roots which you had cut, you have to arrange them and transfer them into freshly prepared acetoalcohol. Nothing, just glacial acetic and acetonol are in the. Which organelle is known as powerhouse of the cell? Very easy, mitochondria. Why? Because it transfers or it supplies energy needed for most cellular activities, and this energy is stored in the form of ATP. Right now I discussed, which is adenosine triphosphate. What is the watery fluid inside a large central vacuole known as cell sap? Which scientist stated that new cells are from pre-existing cells only? Rudolf Virchow in 1855. Name the green color pigment present in chloroplast. Chlorophyll. What is the dense round body inside the nucleus? Nucleolus. Nucleus के अंदर nucleolus. What is the molecule that acts as energy currency? देखो जहाँ पे आप जी energy currency आ जाए या mitochondria आ जाए तो उससे adenosine triphosphate ATP. Name 2 major type of cell division. Major का in your book 2 are only given: mitosis and meiosis. Fine. 2 major and if you want question is of more than 2 marks, you can write that mitosis is for growth and repair, whereas meiosis was sexual reproduction, genetic diversity. And in mitosis you can also see asexual reproduction. Okay. What are colorless plastid that stored food? Leucoplast. Differentiate between the following pairs on the basis of their permeability, cell membranes and cell wall. देखो, it is selectively permeable, allows only certain substance to pass through it, so obviously it will control what enters and exit. Whereas cell wall, it is fully permeable, है ना? It allows most of the substance to pass through, means it does not control. RER, SER. RER means has ribosomes attached on its surface, that's why it appears rough, है ना? Or I want to correct one thing. देखो, RER and SER given. There is one technical error. You can say this whole point is of RER. This second point is of SER. My bad, है ना? So mainly involved in the protein synthesis secretion of RER. Then of SER it is given here no ribosomes on its surface. That's why it appears smooth. Involved in the synthesis of storage of fat and hormone. In the MCQ also it was given. Then differentiate between the following pairs of terms on the basis of the pigment chloroplast, chromoplast. Again here this first point is of chloroplast whole, second is chromoplast. So chloro, it contains green color pigment called chlorophyll found in leaves and green plants. So अगर chromo होगा तभी तो it will be responsible for the photosynthesis, and chromo, they are the pigment that contain yellow, orange or another pigment found in flowers, roots, responsible for attracting colors, responsible for attracting pollinators and pollination. I have explained in my previous video. So this you should remember that why is it important for photosynthesis, and this is for attracting bright colors, they attract the pollinators. Which of the following pair of cell organelle contains DNA? Chloroplast, no. Mitochondria, nucleus, yes. Second option is the correct option. Identify the pair that incorrectly matches. Incorrectly, ribosomes, protein synthesis, it is to be correct. SER, lipid and cellulose. Cellulose होता नहीं है. This one is the correct option. What is the difference between diffusion and osmosis? So देखो, diffusion is the net movement of any particles from a region of high concentration to lower concentration, which occurs without a membranes. देखो, if you look at these definitions, तो somewhat they are same, but a major difference is that in diffusion, the movement of water from a dilute concentration to less concentrated area is without a membranes, whereas osmosis occurs with a membrane, selectively permeable, है ना? So that is the major difference. Why does a potato peel swell up in plain water but shrinks in a salt solution? You just have to explain the difference between hypertonic and hypertonic. Differentiate between endoplasmic reticulum and smooth endoplasmic reticulum and rough endoplasmic reticulum on the basis of their function. We have already differentiated. What is the function of Golgi apertures? So देखो, the Golgi apertures acts like cell's post office. This term is to be remembered. Packaging and shipping center. It receives proteins and lipids from the endoplasmic reticulum. कहाँ से बेटा ये लेता है proteins and lipids? From the endoplasmic reticulum, and then modifies salts in packages into vessels. Why do plant cells shrink in shape when placed in salt solution unlike animal cells? देखो, why? Because plant cells have rigid cell wall made up of cellulose outside the cell membranes. When placed in concentrated solution, water leaves the cell by osmosis, but the cell wall remains in the outer sheet. Only the inner contents cytoplasmic sheet. Whereas in animal cells which lack a cell wall have no such production and shrink entirely when water leaves. What are chromoplasts? What is their role in flowers and fruits? The chromoplasts are plastids. Abhi in differentiation also we discussed, found in flowers, petals and fruits that contain pigment other than chlorophyll, means green, such as yellow, yellow, orange, red and other bright. They give bright colors to flowers and fruit, attracting pollinators for pollination. What is contact inhibition? How does its failure lead to cancer, a kind of tumor? So I have already discussed. Contact inhibition is a process by which animal cells automatically stop dividing when they come into physical contact with neighbor cells, keeping growth controlled. है ना, उसमें क्या होता है? Contact हुआ ना बेटा, so they automatically stop dividing when they come into physical contact with neighboring cells, keeping growth controlled. When this mechanism fails, then the cells lose their control and divide uncontrollably, forming tumors, and errors in mitosis can cause abnormal chromosomes number. Cancerous tumor can be spread. Fine, I hope now it is understood. Okay, So here uh, diagram is also given. First, controlling all the activities, obviously nucleus, site of cellular respiration, obviously mitochondria. Then storage organelle that also provides rigidity, storage organelle, rigidity, vacuole. Then what separates the cell contents from their surrounding. Cell membranes provide structural rigidity. Cell wall, pack, packs and sorts. Packs से और post office से should remember. और ये देखो received from ER. जब भी I received from ER या post office, that at that time only should click in your mind.
Due
Oh
to
yeah,
some glitch, the handwritten
helps in
notes aren't
manufacturing food.
available. So
Chloroplast
I
in
will be
plants.
providing you the links in the
क्योंकि
description
animals
by
तो they
which you can
can't
check
create
them
their
out.
own
There is no
food, they are
need.
heterotrophs.
Everything we have discussed. So
Fine, so this
study.
was
Thank
all
you. Bye
about
bye.
the
Take care.
question.